Good afternoon, everybody. All right, so if you continue from last week, last week we stopped at this question. Okay, something to do with genetic engineering. So we move on to the next question from our first week's worksheet. If you have brought it today, you can turn to that. Now, this is from last week. We haven't finished last week's worksheet. Okay, so if you brought it, it's great. Okay, if you haven't, then just listen in. Okay, so this question is about sickle cell anemia. What is sickle cell anemia, guys? Okay, a recap. Number one, it's when the blood is sickle-shaped, just like this. Okay, it's shaped like a sickle. Why? Because of a mutation, a change in the base sequence of DNA, and then the gene for producing hemoglobin. So this mutation is on the gene for producing hemoglobin. So we know that genes, they will code for amino acids being produced. So when you have a faulty gene, you have faulty amino acids. So this is what's happening, and the hemoglobin is abnormal. All right, so the hemoglobin will form rods and pull it into a stiff sickle shape, just like this. Normal should be looking like this. Oh, y'all cannot see, Okay, when I use the cursor. Okay, normal should be looking like this, where they are flexible. Okay, a biconcave shape. They can twist to fit into the capillaries. But if you have a sickle shape with a cell like this, it will get stuck. And what will happen when this side is stuck? You have a blood clot. And what will happen because of that? Your blood cannot flow down this way. It can flow this way maybe, but not flow down this way. And this area will not have oxygen, will not have nutrients, and it will be painful because of the lactic acid that's building up. Okay, so this part is painful. The person will feel it. Okay, another common question in IGCSE they'll ask is, oops, where did I go? Okay. Sickle cell anemia, what are the symptoms? So you know this, guys? Pain. Okay, we talked about it. When there is a blood clot here, it's painful here. Sometimes they can get pain in their joints because the blood doesn't flow to the joints. The joints are painful. All right, and then they also have frequent infections because not just blood clotting, but when that organ does not get enough oxygen, the tissues can die and the damage is done to that organ. So one of the organs that could be damaged is the spleen. Have you guys heard of the spleen? What is the spleen? Okay, let's go find a picture that shows us what the spleen is. I think you've heard of it, but you do not know where it is, right? Nah, it's right there. If that makes any sense to you. Is it important? Okay, it's to help. It's part of your lymphatic system, where the lymph fluid will flow through, and you have a lot of, you can see my cursor, right? White blood cells waiting in here. What do white blood cells do? Richard? What do white blood cells do? Kill bacteria. So if your spleen is damaged, can the bacteria be killed by the spleen? No. And your spleen is quite an important player in this killing the bacteria thing. So as a result, the person will have frequent infections, get sick easily. Not just that. Okay, anemia. Okay, what is anemia? Ding, what is anemia? Do you know? Okay, Jolie? The red blood cell count is low. Does that mean that the bone marrow is producing less red blood cell in the sickle cell anemia person? No, it's not. But it's because that this kind of cells, right, red blood cells, they die very fast. This one, the normal ones, will last for 3 months. 120 days, about that. Okay, but this one can only last for 10 to 20 days. Means that before your bone marrow can make enough to replenish what is lost, your red blood cells are already dying very fast, faster than what your bone marrow can replenish. So as a result, the person gets anemia, not enough red blood cell. And okay, when the person has anemia, all this will happen, this and this. Red blood cells are for carrying oxygen. So if you do not have enough, feel tired. Okay, do not have enough, your oxygen cannot be passed around in the cells among the cells in your body, so you will feel breathless, especially when you're doing exercise. Okay, not just that, but also when you do not have blood flowing properly through all parts of your body, all right, you're going to have problems bringing nutrition, bringing oxygen, so the person cannot grow very well. That's why we have delayed growth. So can you guys remember this? 1, 2, 3, 4, 5, 6. 6 different symptoms. Then, all right. Of course, one of the symptoms is death. Okay. Your answers, if you come across this question, they always have the word death as one of the answers. Okay, so yeah, you could put that. All right, but if you don't want, you put the first 4, the first 5. Yes. Can I put what? Okay, of course. Of course, a lot of children die from this young. Because, the places where sickle cell anemia is found is also in the places that are very poor, like Africa, certain parts of Asia where they are not very, they don't have very good medical systems. So a lot of parents didn't realize their children have sickle cell anemia. Okay, now as babies, I think a lot of us would, the doctors would take a blood test the moment the baby is born, take a blood test and they'll check any abnormalities. So if there's sickle cell anemia, oh, your baby has sickle cell anemia, we must give them treatment. But there's no cure, okay, for this disease. So the doctor can help to reduce pain, all right, but there's no cure. So sometimes if the blood clot is very major, the person can die at a young age. So they will give blood thinners and other things to prevent this, if the parents know and they send to the doctor. All right. Okay. The next question, important. What's the relationship between sickle cell anemia and malaria? Okay, previously I taught this before, right? You want to remember this? Okay, so how do you answer the question, why, okay, in areas that there are sickle cell anemia, there are a lot of malaria, there's also found to be a lot of people having sickle cell alleles. Is there a relationship? Yes? Okay, they do find, maybe there are other reasons, okay, but they find that generally there's a general similar pattern. It won't be exactly the same. So maybe there are other reasons, but, okay, so the scientists were like thinking, okay, what's the reason? And they kind of did some theories here, and they studied it, and they found that maybe it works, okay? It's correct. So you have to learn it. So I've organized this into a table. In this region, okay, where there are people having sickle cell alleles, some have, some don't have, you have 3 types of people. One is that they are totally normal. They don't have sickle cell alleles. HbA, we call this. A is for normal. Okay, I made the correction from the previous lesson. And then you have people they have two sickle cell alleles. That means they inherited one from their mom and one from their dad. So they definitely will have sickle cell anemia. Okay? And then you have another group of people who have one sickle cell allele but one normal one. Do these people have sickle cell anemia? If you're heterozygous, no, okay? So therefore, all right, let's talk about how does having sickle cell anemia or having this allele, okay, relate to resistance to malaria. Number one, those that are normal, do they have sickle cell anemia? No, but can they die from malaria? Can. So at the end, right, they may die from malaria and they may not survive to have children. So they may not be able to pass on their normal alleles to the next generation. Okay? Now, how about the people who are having sickle cell alleles? Homozygous recessive. Have sickle cell anemia? Yes? But can they get malaria? No. So they're like, okay, we're safe from malaria, but we have sickle cell anemia. Okay? But can they die from sickle cell anemia? May, and they may die young. So they may not have children. They may not pass on their alleles, their sickle cell anemia alleles to the next generation. The third group of people, do they have sickle cell anemia? Do they have? These people, okay, they have one allele for sickle cell and one normal one. Can they have sickle cell anemia? No, they don't have. You must have two of it. All right? So if they have one, no, they won't have. So they're safe. Are they resistant to malaria? Okay, they're safe because they are resistant to malaria. Yay. So, who's the winner in this situation? Who's the winner? Anwen. Who is the one that's most likely to survive both situations? The heterozygous ones. And therefore, this one, guys, survive, reproduce. Okay, these two words always say together. Survive and reproduce, passing on their sickle cell allele to the next generation. So in this case, what is the result? Sickle cell allele will be more common in this area. All right. And then you will find that more people in the future in places that are having malaria, they will also have the higher frequency of sickle cell alleles. All right, enough talking. Now is question 4. Okay, last week's question, if you have the paper. Explain how red blood cells become sickle-shaped. How would you answer this? Anyone has an idea or not? Jinxiang, where's your paper? Okay, never mind, you can answer verbally. Okay, let me guide you. How red blood cells become sickle-shaped? What is the cause of sickle cell anemia first? Say again? It's a genetic mutation, correct. Okay. You can, guys, if you know the answer, you can write down first, then you see whether, you got it. But of course, we're short of time, so I'm trying to rush a bit. Okay, causes of the sickle cell anemia. Okay. Causes, let's change this question. How RBC become sickle shaped? Number 1, genetic mutation. Yes, on which part of the, which chromosome, which gene? Which gene do you think? We just talked about it just now. Is it on the insulin gene? No. Is it on the growth gene? What gene is it? On the hemoglobin gene. Genetic mutation on the hemoglobin gene. Okay. Now, question. Tip, guys. Tip. When you talk about genetic mutation, it's always good to back it up and follow it up with the definition. What is the definition of genetic mutation? Anyone know? Can you raise up your hand? Timothy? Do you have an idea? What is the definition of mutation? Try again. Correct. Change in the DNA. Good. Or change in the gene. Okay, choose one answer, but don't write both. I mean, you can write both. Change in the DNA of a gene also can. This mutation happens on the hemoglobin gene, causing what? So abnormal hemoglobin forms. So you see how to answer this question. This connected to this. This one leads to this. Abnormal hemoglobin is formed. Forming rods. Pulling RBC, the red blood cell. Don't say RBC, say red blood cell. Into a stiff sickle shape. Okay. Basic answer. Of course, there are other points that you could say. Can you see the answers here? Can see? Okay, caused by mutation, change in DNA on a gene for hemoglobin. You say these 3 things, you're safe already, got 3 marks. But do we want to stop at 3 points? No, you want to go more in case your first 2 was not accepted. Okay, the gene of hemoglobin therefore produced an abnormal amino acid sequence or an abnormal amino acid chain. Therefore, abnormal hemoglobin is produced. And what's this AVP? What does AVP mean? Alternate valid points, AVP. So if you're saying something that's related, that makes sense, like this, 1 mark for you. Okay? All right, those people are, okay, I'm going to call out your name. Better be writing the answers. I'm going to wrap this off for the next one. Okay, next one. Some people who have sickle cell anemia have parents, but the parents do not have sickle cell anemia. Remember this question, guys? Parents are normal, right? How come the child has got sickle cell anemia? So how do people inherit the disease? Okay, let's call the girls. Rianne, what do you think? Correct. Both parents are heterozygous for what? What do you claim? Heterozygous for the sickle cell allele. Okay, all right, so you can say that. Okay, let's give the answer for this one, but you can check it later. Okay, both parents are heterozygous for, you must say what? because can be heterozygous for a lot of things. Sickle cell allele. Then what? Rian, continue. Pass on. You want to say, right? Both parents pass down or pass on the recessive allele. Okay, if you know what the name of the recessive allele is, then you put here HBS to their child. So the child will inherit two. Or you could say this, the child inherits two recessive alleles. Okay, making the child what? Child is homozygous dominant, heterozygous, homozygous recessive. Which one is it? Yes. So if you take this step by step, you're more likely to get marks, okay? Recessive. HBS, HBS, and therefore they get the sickle cell anemia. Okay, wait. Therefore suffers from sickle cell anemia. Anyone has anything to add here? I might have missed out something. Because it says 4 marks, but I only have like 3 points, I think. Or I hope, okay, that maybe one of these points are worth more than that, more than 1 mark. So you start brainstorming. Do you have anything else to say? What did they mark for? Okay, this one. Both parents carry the recessive allele. Okay, you can say something like that. Both parents are heterozygous. Half the gametes of both parents have the recessive allele is true. Okay. Because if they're heterozygous, like for the father, if the father is heterozygous, 50% of the sperm is normal. 50% of the sperm is carrying the allele. And then children who are homozygous recessive. And then you can say, oh, therefore there is a 25% chance of having sickle cell anemia. So how we mark for this answer? Both parents are heterozygous. Okay, and then we should add in both parents for the sickle cell allele, meaning that both parents carry, they are carriers. Okay, so this one will be marked 1 mark if you said this. And then they passed on their recessive allele to the child. And then the child is homozygous recessive. And one more point is there is a 25% chance of the child having sickle cell anemia. Okay, sickle cell anemia. All right, did you guys see how it's answered? Step by step. This question is common, right? You've seen it before, right? In another paper. Okay. Next one, let's jump. Now, do you know the answer to this question? Define the term species. Because this question comes up very commonly. Very frequently. Okay, my English is very bad today. Very frequently. What's the answer? Dorcas, do you know? Species. How do we know that the dogs are the same species? And how do we know that dogs is a different species from wolves? Same genus, but different species. The same family, like you can call them the same family, but they are different species. First of all, can a German Shepherd so large maybe have a cross with, I don't know, Pomeranian? You guys want to see like a mixed Pom with German Shepherd? Is it possible? German Shepherd mixed with Pomeranian? Can, you can mix. Okay, we have all sorts. Even Dachshund can mix with, I don't know what other dog breed. But the thing is, they are all the same species because when they mix and they breed, right, their offspring can have children can have puppies of their own also. Right. So what makes two animals the same species is they can, okay, individuals that can breed or that can reproduce, you say breed like better, okay? To produce this one. The offspring can have children of offspring of their own. Then you can have a new line of mixed dog breed. Anyone have any ideas of a nice dog breed that you want to share with the class, that we can have some pictures of it? Now, pit bull and husky. That would be interesting to look at. There are pictures. Pit. They call it what? A pit ski. What does it look like? Does that look like a pit bull to you? Same color. Does that look like a husky to you? Oh, he's got you talk to the parent like, oh, he's got your eyes. Oh, he's got your hair. Okay, so yeah, they mix characteristics from both parents. Okay, and these ones, they can go on to have their own pit ski generation, like the whole breed of it, whole lineage of it. If people like this breed lah. So this is what we call selective breeding. You choose which one you want, you mix them, hopefully you get something nice. Right? Okay, how about this one? We talked about this before, the liger. The largest cats in the world. Okay, wait, how large is it actually? Okay, larger than a lion, larger than a tiger. What's a liger? The father's a lion, the mother's a tiger. Together, right? Okay, it's not easy for them to breed. It's not easy for them to create. You mix a lion, you mix a tiger. You hope they breed, but it's not easy for them to actually have offspring. And if they do have offspring, the offspring survives and grows. All right, that offspring is the last, will not have any more kids. Because it is not, their parents are not of the same species. So each liger will not have children. You cannot have liger and liger mix, new species coming out there. No. All right. These are what we call hybrids, but they're not called species because they do not produce, what's that? Fertile offspring. This question comes up quite a lot, okay? Next one. Explain the distribution of sickle cell anemia as a result of natural selection. Explain the distribution in human populations. How to answer this, guys? How do you answer this? How does natural selection cause change the sickle cells distribution? This question is always tied in with malaria. Okay? It's always tied in with this answer that we talked about just now. There we go. You want to read the answer straight? Okay, cross-check it with what we talked about just now. People with sickle cell anemia are resistant to malaria, or people who are heterozygous, okay, are resistant to malaria, so they survive. But people who have sickle cell anemia will die because of sickle cell disease. People who are heterozygous are more likely to survive. Then when they're more likely to survive, they will breed. Okay, survive and reproduce. Guys, remember this, survive and reproduce. Always say that together. So you can get 1 mark here, 1 mark here. And then always follow with pass on. Okay, we move on to the next one. Okay, so we have the week 2 papers. Okay, what I want to do first for the next set is to go straight to the last question. The one with the shark. About fishing. Okay, so last week we talked about how do we conserve species that are endangered. Do you have this paper? Over there. You take and you clip it on your own. Okay, last week we talked about how do we conserve animals that are endangered, having breeding programs. Can we help also our aquatic life? Right, so they are also being fished and their numbers are growing lesser. And can you imagine if we didn't do anything, the next 20 years your children will have no fish to eat from the sea. All the fish you have is probably from the rivers. Then when you go snorkeling, nothing there, all water only, there's no fishes. Right, you go diving, hoping to see some fishes. No, you don't see anything. It's just empty water. What happened? Okay, it's because humans probably have fished too much, or pollution, or global warming. So we're going to talk about that. Knowing that our fish numbers have dropped down, if you can see also, from this, you can see the numbers are actually dropping down. Okay, I suppose this is an actual graph from actual data. It's dropping down from the year 1990 to 2000. Okay, then it starts to rise again. Well, good. But at this time, right, this is because we're doing something to help the fishes. But if we didn't do anything, it will just go down, 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 down. By 2020, we have no fishes to eat already. No sea fishes. So, question, how do we conserve and manage fish stocks? 3 ways, according to your syllabus. Number 1, how to manage fish stocks. That means the number of fishes in the sea, okay? You are step number 1, guys. We're trying to educate you what to do with the fishes. Don't eat too much fish. Eat more chicken. Eat more vegetables, guys. Don't eat your fish. That is wrong, okay? All right. So there's a certain balance we need to achieve. Number 2, governments will have to issue quotas, okay? Legal quotas. What is a quota? Legal is to do with laws. Quota is to do with limiting numbers. Okay, I'm going to talk about that. And number 3, I forgot already. Something to do with restocking. Yes, that's the word. Okay, so if you're in a shop and then you want to buy a pair of shoes and they say, sorry, no size, out of stock. What do? So, okay, I'll wait for you to, when will the new stock come? Oh yeah, we'll be restocking it next week. That means they'll bring in a new batch of shoes next week. Can we bring in a new batch of fish to the sea? Okay, that's something to do with restocking. All right, let's talk about education first. All right, so we have to educate you why the fish numbers are dropping. Because of, number 1, water pollution. Okay, all the factories are dumping chemicals into the ocean. Maybe radiation, certain, like in Japan, the Fukushima disaster, where the radioactive plants actually was leaking radioactive waste into water. Okay, I would imagine those fishes in the area died a lot too. Climate change, it's getting warmer, and when it gets warmer, fishes cannot reproduce. Okay, or they may die faster, or they will swim somewhere else where it's colder. So you see that the warm areas, okay, will have lesser fish. The cold areas, which is far, far away, has more fish, maybe. Overfishing. A lot of you like to eat fish. It's a good business. They catch so much fish that actually they catch all the fish. They didn't allow some younger fish to continue to survive and reproduce. So the numbers in the next generation is lower. So we have to spread awareness through education to you guys, because you are the consumers, and also among general public, and also among the fishing industry, because they are the ones who are first line in this issue. Okay, what can they do? Well, check what fishing methods you're using. A lot of the fishing methods last time, they used trawling. You can catch a lot of fish. Okay, this is the ship. Then this is the water. They take a net and they drag it all the way to the bottom. Okay, maybe they have 2 ships to do this. Something like this, okay? I'm no fisherman, so I'm not sure exactly. But they will drag the net on the bottom. So everything here will be caught. And then they'll pull it up. And then when they pull up, you imagine they only want certain fishes. Not every fish in the sea we can eat. But they'll catch every single fish in the sea, every single thing, pull it up, and they all die. And they realize, oh, this one I don't want, throw that in the water. Oh, this one I don't want, throw that in the water. But they're dead. Because you really pull them up onto the boat, and they're on the boat for a few hours, then you throw them back. So, while you get to keep the fish that you want to sell, thousands of other fishes that you don't want to sell are dead because of this method. Okay? Another one which they suggest is better is what we call the purse method, like this. Okay, this one you don't bring all the fishes up. What you do? Okay, different. One boat only, so one boat only. You know your fish is swimming, because fishes, right, they tend to swim at certain depths now. Some of the fishes that you want, they're swimming on the top. So you do put out a net like this. Okay. And then it's just like a circle. And then you take a drawstring and you pull it like that. So that what you get in the end, you get to collect the fishes in a purse. Then you send out a little boat over here. All the fishes are still swimming nicely and happily here, wondering what's going on. Then the guy in the boat is going to catch only the fishes that he wants. He's just going to collect all the fishes that he wants, and the ones he doesn't want, he put it back. So in this case, it's actually better for the fishes because you only catch the fishes that you want, but you do not harm so much the fishes that you don't want. Okay, and they say this is what we call the purse netting. Another one, using suitable mesh sizes and shapes. Sometimes the fish you want to catch is very big. So don't use, if you want such big fish, right, you just catch you don't want a small fish, you need to allow the small fishes to escape. So you use the net size that is enough to catch the big fishes, but big enough to let the small fishes swim through. That will be a suitable mesh size. And then, not just that, mesh shape also is important. What shape is this? Diamond. Okay? This is not good for your fish. Why? Because this does not allow small fishes to swim through. The diamond ones, when you pull it up, they will actually catch all the small fishes also. So, not recommended. The diamond mesh. Okay? What's recommended is the square mesh. This one will allow small fishes to swim through, but catch the large fishes that you want to sell inside. So the baby fish can swim out, the young fish swims out, and the young fish will continue to grow and lay their eggs so that the new generation is there before they grow big enough to be caught the next round. So by having the square mesh, you allow the young fishes to escape, only catching the adult fishes. So this one will allow you to catch and the fish that you want at the adult size that you want and reduce the trash fish. Trash fish means that the fish that you don't want. Okay, you don't catch them too much, and also the next year when you go back to the sea, the baby fishes that escape, now they grow big. Now you can catch them. So you do not actually cause the fish population to drastically drop. You actually give them a chance to go escape and reproduce. Okay, another one, governments will issue legal quotas. Okay, 3 ways. They have to issue licenses that will limit the number of people that are going out to catch fish. Say that maybe 1,000 people want to go and catch fish. Anybody can just go there and catch fish. Like that, right? The whole fish population will be wiped out. So you make sure that you only issue licenses for like 50 companies, 50 boats to go out. So when these 50 boats go out, they can catch their fish, all right? But they can't catch so much also. So you get to control how much fish is caught. Not just that, limit the areas for catching, for fishing. All right. So you cannot, actually, if you're a fisherman, you cannot simply just go as far as you want and catch fish. It's illegal. You're only allowed to catch within a certain radius from your country. I'm not sure what country this is. Okay, but you can see that this country has a certain radius where you can only catch fish from in this area, and you cannot cross into the next country's waters. So this way, the governments can help to control the number of fishes that are caught. Okay, now what about this area? It's what we call protected. That means people cannot go there to fish. So this is protecting the fishes that can escape to this area. So fishes grow, lay their eggs, okay, replenish numbers, then they can swim back here, and then they get caught next year. So this is one of the ways that they manage how much fish that people catch, and also manage the size of catches. Probably you're only allowed to catch, I don't know how much, 1 ton of fish. When you caught your 1 ton, 1,000 kg, is it? Of fish? You gotta come back already. You cannot go out anymore. Okay, so you cannot be so greedy and catch so much fish. So, do you think the fishermen are happy about this? No, right? So they say that, since you are creating all these quotas, you got to help the fishermen in other ways. The government's supposed to help them to, maybe be able to buy other things cheaper, subsidies, or give them training to go into agriculture, so in other areas, right? So to help them because their livelihood is being affected. Third way, fish stocks. Remember about the shoes, not enough shoes? Next week they come with a new batch. Same thing here, okay? So not enough fish, it's okay, all right? We will help the fish. We collect a few, bring to our farm, give them lots of food, make them really happy. They have a lot of babies. Bring them back to the water, let them go again. So we can do this for lakes and rivers. It's easier. But for sea, it's a bit difficult because it's too big, right? So they do a lot of restocking for rivers and lakes. All right. Valuable fish are bred in breeding programs. Release. Okay. Why am I saying all this, guys? Because this is actually part of the next question. Okay. This has come up for IGCSE before. Okay, if this question came up for IGCSE, would you know how to answer this? So these are the answers. Okay, so first of all, in restocking, what happens is valuable fish are bred in breeding programs. Then you release them back into the wild. 2 places. Either you go back to the wild or you release them into protected areas. Right? So in the wild, they can be caught. In protected areas, they don't get caught. So that they continue to grow even more. Fishing is not allowed in protected areas to allow the fish population to grow. So you have people monitoring how much fish there is. So you have somebody taking care of the fish while they grow. All right. So while they grow, it doesn't happen overnight, How much fish can you eat in one day? How fast does it take for you to finish one fish? 10 minutes? Okay. How long did that fish take to grow to that size? A few months, So if you're eating you are eating at such a high rate, the fish takes 1 month, 2 months to grow to that size. You gotta wait, gotta slow down, let them grow first. So this is what they do. Limiting you eat other things first, other types of fish while waiting for this fish to grow larger and hopefully have offspring of their own. Then only you eat them. Okay, and meanwhile you cannot fish too much also. Don't catch them so fast, catch them slowly. Don't catch too much in one day. One day, I don't know what, 1 kg. I don't know how much, 5 kg of fish. So you allow your wild stock to recover. 1, 2, 3, 1, 2, 3, 4, 5, 6. Can guys? Okay? Okay. Are we ready to go into the... Oh, wait. I got another picture. You know what this is? Can you guys tell me what is this? These are fish. This is a plane. You ever want to jump off a plane? Okay, why? They are trying to restock the fish in a place that's very hard for them to go to. Okay, they want to bring, when you bring fish, it's not just you bring 10 fish and go there. No. You have to bring thousands of fish, carry it all the way, I don't know, go up the mountains, go through the jungles and everything, finally reach this lake and let them go. On the way, I don't know how difficult it is to carry all the fishes and lug them all up to the mountain and all that. So, take the plane. You're surrendering the fish to their death. So apparently, they did studies that some may die lah. Some didn't swim, some didn't dive this way. Some dive, I don't know. Okay, when you dive, you have to dive this way, right? You cannot dive flat, right? You can die, right? So the fish actually goes down this way. So the fish actually just goes zoop into the water and swims away. So not many of them die. Okay, one of the ways to restock your fish plane in areas that are hard to reach. Question 7, guys. Papers. Okay, look at the graph. Study the graph. When you study a graph, first 2 things you must look at. What are they? Sometimes you can see it from here, from far away. Can you see? Can you see it? Okay, what are 2 things you must look at when you look at a graph? The first 2 things. Do you all look at the line first? No, right? You always look at not even this one now. X, Y, then only the line. This one tells us the years from 1970 to 2010. This one tells us the millions in tons, million tons, okay? 1 million, 2 million, 3 million, 4, and 5, 6, 7, 8, 9. Atlantic cod, this one. Read the whole sentence. Read the question. "1970, the mass of Atlantic cod was 8 million tons. 1970, 8 million tons. Okay, correct. State 1 year when the mass of the cod was half the value of this 8 million. What do we do? See this? There are few places that you can get it, like it hits 4 million. The few years. 1, 2, 3, 4. Okay, choose one of them only. $1995. Am I right? About 1995. It could be 1996. How do we know how to read this? There are 10 boxes. Five years. If you want to count one box is how many years, you put the box at the bottom. You put that number at the bottom of the fraction. So we see that one box has got how many years? 0.5. Okay, my pen is not with me. Per box. Okay, next time I will use my pen. All right, so 0.5 per box. All right, that's how we read the 4 is here. This is like 1995, 1990, okay, 1990, almost 1996, 1997, you can choose that, 6, 7. What year is this, guys? It's right in the middle. What year is this? Okay, so this is how it's in 2006, 2007, 2007 and a half. So it's 2000 and year 2000, yeah, 6, 5, 6, 7. It's year 2007, this one. There we go. Choose one of the years, okay? Next question. State the years when there's a continuous increase of the mass of Atlantic cod. Okay. Have a look at your papers. Choose the 5-year period. That means you must say from what year to what year? Dean, what do you think? A bit risky because 1975, right, is actually going down after that. Give the exact year. You can say 1975, but you see here 1975, it's actually going down. Then only it goes up again. So I'll be playing safe and say, don't say that. Can you say something else? What other year? Okay, 1976 to 1981 plus 5. If you start in 1976, then you finish at 1981. Here, continuous increase. No other years, no other region here where you have a continuous increase. Except you want to say, okay, you can't say 2005. Did anyone think of saying 2005 to 2010? Risky. You know why? It's not continuously increasing. It is remaining the same constant and then only increasing. So don't say that. Okay, so the best is actually within this time period, 1976 to 1981. Or if you want to go a bit higher, 1984, 1979 to 1984. Okay? Okay, so this is how you read the graph. You realize that in 2020 onwards, our papers that I've been doing, they've given you very funny graphs to look at. They're asking you to analyze it in many different ways. So practice, 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 okay? The question of the hour. How fish stocks can be conserved by restocking? What's your answers? Write that down. What did we talk about just now? Try the answer. Number 1. First of all, we take the fish and we do what? Simi, what do we do? Breed them in? Before that, in breeding programs first. You can say protected areas, but the answer they're looking for is breed them in breeding programs. Breeding programs, they make sure that the temperature of the water is right. Nutrients are a lot. Okay, so everything is good for the fish, then the fish will just reproduce a lot. Same like for the panda, they have breeding programs also. Make sure that everything is perfect for the perfect conditions for them. Okay, next. You have the numbers that you want. You have 1,000 fishes. What do? You can release them into 2 places, either into the wild or in or into protected areas where fishing is not allowed. To allow these to grow even more, okay? 1, 2, 3. Safe to say we need one more point. What is that one more point we can say? Isaac Young, do you remember? Try, remember, recall? While waiting for the fish to grow, what do? Do we need to eat only this fish? Like, I don't know what fish is, mackerel. Or what kind of fish is I'm not a fish person, so I'm not sure, not so like into types of fish. Mackerel, there's dory, there's, I don't know what else, no? Shark, not shark. Eat other fish, okay? Let's say. All right, so you can fish other farm fish or consume it. Eat other farmed fish. That means, right? You see, farm fish will be freshwater fish, okay? So it's harder to farm seawater fish, isn't it? So before you eat the seawater fish, while waiting for them to grow, you eat the farm fish, the freshwater ones. That's why they say, oops, sorry. That's why they say certain regions, they cannot get certain seasons of the year. Certain seasons of the year, they cannot get the fishes. Oh. Thank you. First time it happened. I thought some other big problem. Okay, now we know. Just touch first and see. Okay, so next question. Wait, no, answer. This question is weird, not weird, it's rare. When it came out in 2020, I can say that a lot of students were caught off guard because they didn't think that this was an important topic, right now, you guys? So if 2022 November we come out with this question came out, October, November, this question came out, do you think an answer? So now, okay. All right, this is what they said. Exactly, breeding programs, release them into the wild protected areas. When you say protected areas, they actually give you one extra point now. Okay. Monitor the numbers. Use farm fish, wild stocks, recover. AVP, any other suggestions that make sense? Yes. You can be given 1 mark. So your examiner will go and research. If you come up with an answer that they haven't heard before, they'll go find out for you. Okay, they look in the right places, they can find your exact answer. If they can't find it online, then you may lose your mark there. Okay, so next one. Suggest 2 features other than eyesight visible in this. Okay, when they say visible, guys, make sure it's visible in the picture. Suggest that great white sharks are efficient predators. Okay, any suggestions? Isaac Pham, what do you think? How do you know that the shark is the efficient predator? Yeah? How do you know when you go swimming that this fish is not, it's a harmful fish, not a harmless fish? Especially you're going diving there, like, oh, fish, so big, so nice. Shark teeth, okay, that's one of it. Okay, good. And what else? It's huge, is it? Let me see whether that's accepted or not. Who knows? Sharp teeth, yes. Give other answers besides big size. Short teeth. 1. The mouth is large. If the mouth is not large, then it's not out to eat other animals. It's just eating fish or eating grass or something. Okay, just kidding. You realize that herbivores have small mouth. Carnivores have large mouth. Okay, this one has large mouth. Okay, be careful. Okay, let's look at the other answers you can give. Very interesting because yes, large teeth, lots of teeth. They've got rows and rows and rows of teeth, okay? Big jaws, I tell you, if you don't know whether this shark is dangerous or not, you look at the mouth, so you're scared already, okay? Streamline, what is that? How come the shark can swim so fast? When you look from the top, what's the shape of the shark? This one we call a streamlined shape. How does having a streamlined shape help it to swim? Now that you saw this shape, like, oh, okay, good. Now we're going to copy this shape and make it the same shape for ships or planes everything that needs to go fast in water or in air. It's because this is a streamlined shape. Okay, same that fishes have it, sharks have it, birds also have it, except the sunfish. What's a sunfish? Ocean sunfish. That's not a sunfish. That's not a sunfish. Okay, this fish doesn't swim very fast, right? Hey, this fish is huge, the ocean sunfish. Okay, we'll go into that later. You see, it's very small, right? Actually, it's as big as us, It's as tall as us. Yes? Yes, go. All right, so this one. Wait. Suddenly, the internet for me is not working. Okay, wait. Anyways, next question. Camouflaged. How is the shark camouflaged? You know that if the shark's above you, look up, what color is the bottom of the shark? See the picture, what color is the bottom of the shark? White. It's actually camouflaging against the sky. When you're under the water, you look up, it's all white one. So some fishes cannot see the shark coming because it's also as white as the sky. And then when you're looking down, maybe you're a fish that's from the top and the shark is at the bottom. What color is the top of the shark? Gray, dark gray. It's actually almost the same color as the sea water if you're looking from the top. So your fish is two-tone. It's for camouflage. Either you look up at it, you cannot see, you look down at it, you cannot see also. Okay? All right. Next one. Five-mark question. Answer. I got this all from one paper, 2020. Imagine all these questions, right? Came up. How do you answer this? How the ancestors of the great white shark developed adaptive features. What adaptive features? And yes, remember survive and reproduce? So adaptive features are features that allows an animal to survive and reproduce better in the environment. There's a whole definition for this. to better survive and reproduce in their environment. The question is asking, how did this shark develop this feature from the ancestors until the one that you see in the picture? How do you answer this? Any ideas? Okay. Talking about ancestors, adaptive features to today's animal, you're talking about natural selection. Do you remember natural selection, guys? Remember we talked about natural selection. You describe this, you have to say these 6 things. 1, 2, 3, 4, 5, 6. Let's go through them again. All right, so you remember the story about what's this animal? The peppered moth, okay? The type of moth called peppered moth. Variation. First of all, this is the same species, but just different color. So this is considered variation. One is, can you see that? Can you see that? This one? Can you see it? Camouflaging, right? So this is an adaptive feature of the peppered moth in this area, the lichen. You know, sometimes you go to a forest, the bark, the tree, like, is covered with something green and white. In very cold areas. That's what we call lichen. So this allows them to camouflage. Okay, this is an adaptive feature. Okay, coming back to this one. There is variation within the population, such as different color, speckled or black for this one. And you can say this, guys. Variation is caused by mutation. When you say mutation, what do you say? Change of DNA. You detail it, okay? You explain more. Okay, next, production of many offspring. Okay, so one moth will not just produce one offspring, there'll be many, and within this many, there'll be variation. So because there's so many, what will you all do? What will all the offspring do? Compete for resources. And this will cause them to struggle to survive. So who will win? The next part of the story is who will win? Individuals that randomly, not that they develop it on their own, they go training and then they can camouflage better. No, or they go and run and practice their running skills and then they can run faster. No, they don't think about that. Okay? But naturally, randomly, they are better adapted to the environment to survive and reproduce because of their variation. Their variation allows them to be better adapted to survive and reproduce. Next one, passing on the alleles to the next generation. Okay, so in this case, if this area is full of trees with lichen, this peppered moth will survive. It has the advantageous characteristic. Okay, it will go on to reproduce. This one will be eaten by the birds. No more already. So the speckled moth will pass on the alleles to the next generation. More of the future generation will be the same color as this. In another forest where they have a lot of air pollution, the trees are black color because of all the smoke and the smog and the soot. So in this area, the black one will be the lucky one. This one will be eaten. This one will, yay, go on to have children, have offspring, and next generation all black color. Maybe more of them are black, okay? Because black is actually a homozygous recessive trait. Are you able to answer this question? How the ancestors developed the adaptive features? Okay, just now we talked about it. How do we answer that? Do you say variation? First of all, some sharks have better eyesight, some sharks have X-ray vision. Okay, sorry. They cannot see so well. This is because of mutation. Maybe some sharks can see better in the dark. Some can see better in the light, in the when it's daytime. Some can see far, some cannot see so well. Okay, because of mutation. And mutation will, okay, if you want to say more about mutation, it will create new alleles that increase their fitness, making them better competitors. Now, remember the survive and reproduce? Talk about this, the trait. Organisms with good eyesight are more likely to survive and reproduce, passing on the alleles. When you say this, you already have 2 marks ready. Okay, so eventually these alleles for good eyesight, maybe in the daytime or the nighttime or when it's dark, depends, okay, what is required in the environment itself. They will pass this on to the offspring. All or most of the organisms in this population will have good eyesight. So in the case of a shark, right, they suspect that it's because of this. Why they have eyesight? Because not every shark is born with good eyesight last time, but now every shark is born with good eyesight. And talk about this. Always come in conclusion, this is a form of natural selection or evolution. This one is always 1 mark. So in a situation where I do not know how to answer this question, what do? Not sure, right? You underline. There's such thing as adaptive features. What is adaptive features? Features of the individual that, okay, we talked about this, allows them to better survive and to reproduce in the environment. This one, you saw this, right? 1 mark. If you're not sure, you can write this down, you can save yourself. Then you continue talking about passing on the alleles, 2nd mark. Next time, the future generation have good eyesight, 3rd mark. Okay? This is natural selection, 4th mark. Okay, almost 5 marks already. You can save yourself if you do not know what to say. Cannot, guys. Of course, if you think about this, the more practice you have in past year papers, the more exposure you have to different questions. The questions all have the same set of answers, you see. So the more questions you learn, the more of the answers that you learn, the more you're able to answer questions in your IGCSE October, November 2022, okay? Okay, next one, mitosis. You remember mitosis? Are your brains okay for mitosis? What's mitosis? What do you understand from mitosis? The picture is here, but what do you understand from this picture? What is happening here? What's happening to this thing? Something to do with splitting. Okay, what part is splitting? Not to say splitting so much, but what part is splitting? Dave, see you. Okay, so what did Sean say? I'll say again. Split, and it's split into 2. Before that, before they replicate themselves. Yes, before they split into 2, they must make copies first, ma. So this is what's happening here. They will, from this stage, make copies of themselves, then they will split. Okay, let's talk about it. Okay. This one. Number 1. You don't have to remember all the phrases. Okay, A levels you will have to. Good luck to those. You don't have to remember, but understand what's going on because you might be asked about what's going on. So, first of all, when you see this is before mitosis. 80% of the time, the cell looks like this. You cannot see the chromosomes because they are not condensed. They are like very thin threads. You cannot see them through the microscope. But during this time, they know they want to replicate. But if they want to do mitosis, they will duplicate themselves, make a copy. Remember how to make a copy of themselves, guys? A, T, T, A. Remember this, guys? We did this before. This one, whenever this question comes out, I'm very happy. Everyone gets it right. So genes are read in threes, so I must have 6 over here at least. 3, and then this another one. T, G, T, A, okay. When they want to replicate or duplicate, they will split. The DNA's 2 letters will split. Then new bases will form here and form here. Okay. So remember the rules. A must match with, what is it? A must match with T. G must match with C. A matches with T. T matches with A. G and C and T and, okay, this side now here. T and, now look at this, this one. T, C and G, G and G and. Did you get to the same? Okay, so this is replication of DNA that is happening at this part. So you will have this done. When you're ready, move on to the next step. They will condense. Okay, so the thread very long, you will like rub it. That is all like one thick piece like that. So it's this. From this one becoming they're actually very 2 long ones and then becomes like that. This is the chromosome that you always see in the pictures. Condensed form. Condensed into 2 sister chromatids but joined at the centromere. What's a centromere? Let's draw this one here. What's this? What's this? What's this? One only? No chromosome? Chromosome is the whole thing, chromosome. But one only? Chromatid. It's like a kid. Kid chromosome. Chromatid. I don't know. Okay, chromatid is a sister. I don't know where's the brother. Sister chromatid. This one? It's in the center. What do you call it? Centromere. Very important. Because later, the spindle fibers will want to connect here. The attachment site for the spindle fibers. Okay, next one. Then the spindle fibers will form. From the centrioles, they will come and they'll connect to the centromere, connect, attach to the centromere. What will happen next? Line up. So these spindle fibers will pull and then arrange everything in the middle. Spindle fibers pull chromosomes to align at the equator of the cell. What happens next? This is long, so we'll shorten it. Shorten, what will happen when you shorten? This one shorten, this one shorten. The chromosomes will be separated. The centromeres will be broken. Okay, so spindle fibers shorten, pull chromosomes apart. Sister chromatids separate and are pulled to opposite sides of the cell. Yay! Almost done. Cytoplasm will divide. Nuclear membranes will form. So the nucleus, where's the nucleus? No, don't have, disappeared already. Now they'll start forming again. Nuclear membranes will form. Then you have how many daughter cells? How many daughter cells? Santosh. How many daughter cells are formed at the end of mitosis? 1, 2, 3, or 4? I think I cannot hear you. How many daughter cells are formed at the end of mitosis? 2. Are they twins? Are they identical or are they non-identical? Are they exact copies or not? Are they different from each other? Yes, okay. 2 identical, genetically identical cells are formed. Right. Meiosis is different, right? Because meiosis is twice. That means, right, mitosis, but a few differences, and then it happens 2 times. So, I'm not going to talk about this in detail, but I'm going to talk about this one last part here. Xavier, how many daughter cells are formed at the end of meiosis? How many? Okay, again. Beginning, line up, split, cell divide into 2 cells. That's not the end. Part 2. This one will do the same thing. This one will also do the same thing. Line up, split, cell divides. So this one will divide into 2. This one will divide into 2. How many cells are there in the end? 4. Are they identical? What's this? No, they are not. They are not identical, okay? They are not the same. That's why we want meiosis to happen. Because we don't want identical cells. We want variation. So that some gametes will have strengths and some gametes will have other strengths. So we want variation through meiosis. Now this one's important, guys. Comparing between mitosis and meiosis. First of all, mitosis, what happens in the end? 2. Okay, there are 2. Okay, 1/10 to point, 1 finger to hold. 2 genetically identical, yes. 2 genetically identical, haploid or diploid? Diploid cells. What's diploid? The same, that means there are 2 sets of chromosomes. Okay, the same chromosome number as the parent cell, 2n. Meiosis, yeah? In the end, how many? Hey guys, if you can't remember mitosis, meiosis, at least memorize or remember this, okay? How many daughter cells in the end? 4. Genetically identical or non-identical? Non-identical, good. Okay, different. And then, haploid or diploid? Haploid. Say this, you get 2 marks, okay? If you compare these 2, at least 2 marks, I think. Just remember this. Then chromosome number. This one is same. If the parent has 46 chromosomes, they also have 46 chromosomes. But for the sperm, for the ovum, for the pollen grain, for the ovule, if the parent has 46 chromosomes, how many chromosomes does the daughter cell have? 23. Only. So that's what we want. Next, what is mitosis for? For growing. So you need more cells. Okay, grow. Mitosis. You gotta cut. You want more. You want new skin. Okay. Mitosis. Repair of damaged tissues. You have the bacteria. You want to do asexual reproduction. Okay. Mitosis. Okay. But if it's sexual reproduction, you want to produce gametes, which one do you choose? Meiosis. Okay, guys? So, all this, there's a special nickname for mitosis. Copying division, because it's copying exactly for the daughter cells. The special nickname for meiosis is what, guys? Reduction division, because what is being reduced here, guys? Okay, what is being reduced? That's why it's called reduction division for meiosis. The chromosome number is being reduced, yes. Okay, enough talking. Next is your turn to answer the questions. Try these. These are actual cells, guys. Actual chromosomes, they're not drawings. All this while I've been looking at drawings. Before mitosis, at the start of mitosis, they look like this. They're already condensing. Then they are lining up at 34 minutes. Well, it takes them a long time to line up. Sounds familiar now, guys? I don't know. If you got you guys to line up, I'm not sure how long it'll take. 34 minutes. Okay, then after that, split 40 minutes. And then reach the end. Wow, 5 minutes are so slow. It's like move slowly. Let's move to the other side of the cell. 5 minutes later, we'll reach there. Okay, 5 minutes, we'll reach there. Here. I don't know which one's slower, the sloth or the chromosomes. The cell is very small, you know, it's like tiny. Okay, and then 10 minutes time, cell membrane says, guys, you're so slow. I have to do faster. 10 minutes, I will split into 2. Okay, done. State the change that has occurred to the mass of DNA immediately before mitosis. What is that? Okay, let's look at it. Can anyone answer this? State the change that has occurred to the DNA before mitosis. Here. Mass. What they're asking. So if you're not sure what they're asking for, underline the important words. Change. You got to talk about change. Of what? Mass of DNA. Oops. Sorry. So if you don't understand, you underline, and then you answer exactly what you underline. When? This one's important. Remember what we talked earlier? What happened before mitosis? What's happening here? Okay, anyone can give me an answer now. Desiree, do you know the answer for this? Okay, let's look in our slides again. What's happening before mitosis in this place? Chromosomes uncondensed, that means they're not rolled up. Chromosome duplicates itself. One of you, suddenly there's two of you. What will happen to your mass in total? Increase by how many times? Twice. That's right. Interesting. Suddenly, you can double your mass. State the change. Okay, answer is doubles. So you can only say doubles and you get 1 mark. You see something else, they don't accept it. Or it becomes twice. Increases can accept it. Okay. If someone say no change, sorry, wrong. Next, estimate the time when the chromosomes begin to separate. What's this question asking for? Underline important things. Time, that's what I need to tell. Time can be in minutes here because everything's in minutes here. Chromosomes separate. When did they separate? Begin or after? Okay, begin. Then you start thinking, where did the separation happen in the first place? Where did the separation happen, guys? Andrea, do you know? When did the separation happen? Here? Somewhere here? Somewhere here? Somewhere here? Ah, okay. Alright, so remember they lined up here, were they separated already? No. But when they lined up, then the strings pull them apart. Okay, then only they break. They're separating here. So the separation happens between 34 minutes to 40 minutes. Did you guys analyze that? So what answer do you give? Andrea, what answer would you give? 35 minutes can? Can we say 38 minutes? 40? 40 minutes is separated already. Okay, so 40 minutes you cannot because they're already separated. So it has to be between 34 to 40. Sorry, 34 to 39 minutes over here, guys. One number, okay? When it says estimate the time. Now, if a student said, I think it's between 34 to 40 minutes, it is not correct. If they say between 34 to 40 minutes, cannot. You have to say between 34 to 39. Or you just say one number only. One number is safer. Okay, next. With the question. How nuclei or nucleus, many nucleus in cells produced by mitosis, okay, this is what they're asking for. Underline if you don't understand. Good to underline actually. It helps you to understand faster. You don't waste your time. How much time do you have for your paper 4? How much time do you have for your paper 4? 1 hour. 1 hour to do your work, 15 minutes to check, guys. 1 hour, 15 minutes. What can you do in 1 hour and 15 minutes on other days? I think you watch one movie, half of it, that's 1 hour, right? 15 minutes. You watch YouTube for a few YouTube videos, then okay, I don't know, 1 hour and 15 minutes. Very fast, huh? So you have to finish 6 questions plus A, B, C, Ds for everything. It'll be helpful if you underline especially paper 6 also. It will help you to underline important information. It will help you to read faster also, understand faster. All right, so how does it differ from the mitosis? Okay, answer, you guys talked about it. Remember the table that I put up for you guys? If that was the answer, write it down. Okay, those online also, I haven't addressed you till now. Try this question. This is one of the common questions when they're asking about mitosis or meiosis, how they differentiate from each other. Did they take careful? What does the question ask? They're not asking you what is produced at the end of mitosis or meiosis. They're asking you how does the nucleus in the daughter cells at the end differ from the nucleus from the mitosis one. Just talk about the nucleus only. Come, I'm going to come and see answers. I'll come and read some of your answers also. Come. I see you finished already. The question. The paper, the answer questions right here. This question. Finished? Where is it? Which question? This question. My first question. Whatever you remember from just now.
I remember just now is I did the front page only just now. And I did some questions after you skipped earlier.
Yeah, the table that we talked about just now, the differences between mitosis and meiosis.
The reason that I can do it was very
Yeah, but you were talking to them. Haploid, diploid, chromosome number, all those things. Those are comparing between the nucleus. So that. Talk about the chromosome number. Chromosome number, you can explain what diploid haploid is. Talk about the genetic similarities. Those are also important. When you talk about meiosis, you have to talk about mitosis, whereas mitosis is this. Always compare between the two. Where? The number of chromosomes have been halved where? In meiosis or mitosis? Yeah, so you have to, even though the question is phrased like this, but you have to be specific. Right, so you put that in. All right. Look at the answers. 3 marks, only 3 answers are given. That's very risky. Usually 3 marks, they give you a whole list that it can be accepted. This one, you got to be spot on, okay, accurate. Oh, sorry, Richard. I'll make it larger for you. Okay. Sorry. Okay. Number 1. It's because we're talking about meiosis. I think you better mention meiosis. Meiosis, daughter cells and meiosis are genetically different, whereas daughter cells and mitosis are genetically identical. It's better to say both. Okay. In meiosis, daughter cells are haploid. In mitosis, daughter cells are diploid. You should mention both. Haploid, and you say the meaning. Because whenever you talk about haploid, guys, always talk about the meaning of haploid. Trini. Okay, so tell me, when it's haploid, what does that mean? What does that mean? What's 1? Talk about the chromosome number is half. Or you can say haploid is 1 set of chromosomes. Okay, you can say that. But they're also looking for, in meiosis, chromosome number is being halved. Whereas in mitosis, the chromosome number is maintained to be the same. Okay, write this down. Common question. Next one also common question. Starting to ask, what's stem cells? You know what stem cells are? What are they? I don't want to show you the answers. Okay, come. Let me hide it. There we go. I'll be smarter now. Okay, stem cells divide by mitosis. What's stem cells' role in the growth of an embryo? What's a stem cell? Always this main word there. Specialized or unspecialized? Unspecialized. What can they become? Unspecialized cells that can develop into different tissues. Okay, so 2 marks, right? Okay. Stem cells, unspecialized cells of a zygote, of an embryo. Yeah, I don't say that because sometimes you get stem cells elsewhere. So that can go on to develop into specific specialized cells. Okay, I think my answer is going to be wrong. I think there's something else to say. Try that. Julie, what do you think? What's the answer? Just now you pointed to it. Yeah, we can talk about it. Okay, you can say mitosis. But this one, in this case, they do not give marks for that. But you can talk about that. That is correct. Okay. Stem cells are unspecialized cells that can go on to become specialized cells. Okay, why? All right. The question is, you know, our eyes, okay, the cells in our iris, right, has the exact same chromosomes as the cells. Is there another class after this? No, I'm just checking. Is there another class? Oh, okay, so I should end here already. Our eye cells has the same cells as our skin cells, but how come our skin is not having the same characteristic as the eye? That's because, right, even though they all have the same genes, sorry, they have the same genes, but not all the genes are expressed. So that's what it's saying here. even though they all have the same chromosomes, but only certain chromosomes are working in certain areas. Expression of genes in specialized cells, certain genes are only expressed. Okay? All right. So we pause here. Thank you very much, everybody. Thank you very much, those online.